Okay. All right, well, hi everyone, and thank you very much for the invitation to be here. Today I'll be talking on a slightly different topic than my title said. I'll be talking about single molecule imaging and going into this idea of superlocalization that WE introduced earlier. But I'll be beginning by talking about bacteria imaging, and in particular one example of 3D super resolution imaging that we did in Caulobacter crescentus when I was at Stanford. And then I'd like to switch gears and talk about imaging highly porous solid state materials, because it turns out that there are other interesting systems than just biologicals. Though I'm very excited about bacteria cells, these materials have highly heterogeneous diffusion that really begs for study on the nanometer scale. Before I begin, though, I thought I really should motivate, sort of why we're all here to celebrate WE. And there's a great legacy from his lab of careful experimentation, also just a very nice collegial place to do good science. But as well, something that we learned from WE was to always have a plan. And so everything's very organized in the Murner lab. In particular, if we're moving equipment, and when I arrived at Stanford, we actually had an issue of having to move a laser about 12 times up and down a stair with no elevator. And so over here on the left, you can see WE, and he's directing traffic and making sure that these seemingly smart scientists can actually carry this very expensive laser properly. Additionally, WE was a very hands-on advisor. One of my favorite things about working at Stanford was that if you can't find something, and this something could be as small as a wrench, WE, as the PI, was actually the person who could find it. And so here he is doing some electrical work. Finally, the last legacy that I learned was just to be organized. And something when I met Callie for the first time, actually 2 years ago, was talking to Callie and to Jim Shuck, who also has a lab now. And we were talking about how in all of our labs, and probably all the Murner alum, Randy's nodding as well, all the Murner alums have filing cabinets where everything must be found, and every drawer is labeled. And these are not pictures from the Murner lab, these are pictures from my own lab. And in particular, I want to point out this part. This is the drawer for the label maker. So the other thing we learned was that we should never, ever, ever average over ensembles. And there's some joke to be made here about how this is actually not the same baby. Oops, this is two different babies, born 3 years apart, imaged in different states. So as has been mentioned in the last two talks, the way that we use single molecules to beat the diffraction limit is by imaging the molecules one at a time. And WE, in a review about 5 years ago, likened this to looking at single mountains. And it's very hard to find examples of single isolated mountains. I like this one. This is Mont Saint-Grégoire, near where I'm from in Montreal. Just an example of a single mountain. And no one in this audience would think to tell me, even though this mountain is kilometers in diameter, that the peak may be over here. You could tell with very reasonable certainty that the peak is somewhere over to here. And the accuracy with which you could find that position is basically a signal-to-noise problem. It's the difference between a smooth mountain and a craggy peak. Well, what we do in single-molecule imaging, of course, is that we fit a 2D Gaussian to each single mountain. And the precision of the determination, again, depends on signal-to-noise ratio. Nanometer scale localization accuracy is achievable. And the important thing is the resolution with which we can find that center is much better than the whole width of the mountain. And so this is just a mountain, and it's very large, but what we like to do is mostly look at single molecules in cells. And so these are Caulobacter crescentus cells. This is work that was done by Soyoung Kim before my time in the single molecule world. Here, this mountain that you're seeing is the fluorescence coming from a single YFP, a single fluorescent protein molecule that's attached to a structural molecule of interest. This is the real data that we see on a CCD camera, and this is a sexied up version where the height and the color just give you the fluorescence intensity. But in either case, we could say with high level of certainty where the position is by fitting it. So we get high resolution information, and the reason I think this is very important is that we also get dynamics. And so this is but a single frame of a movie, and here you could see this molecule of interest doing a zigzag back and forth. This movie's been accelerated 100-fold. The motion's actually 6 nanometers per second. And it's really this paper that got me excited about single molecules that are read right before moving to Stanford. So there's 3 principal things that people like to do with single molecule imaging. Once you've found your molecule and found its position on the nanometer scale, the first thing we could do is track single particles. And I'll give you an example of us doing that in microporous materials later on. The second thing we could do is 2-color imaging. Even though these 2 mountains are not isolated, because they're in different color channels, they're overlapping. So rather than being spaced out in time, they're spaced out in color. And finally, there's this idea of doing a reconstruction, a super resolution reconstruction, where, as W.E. illustrated before, there's some underlying structure, and when we look at a few molecules at a time, over time we could build up a map of the localizations. And these have a lot of different acronyms. In the murder lab, we like to call that SMACM, or single molecule active control microscopy. And I'd like today to tell you an example of using dynamics as the active control mechanism. So why are we interested in looking at live bacteria cells, which is the heart of my postdoc work and my lab at Michigan? I like to tell students that bacteria cells are exactly why I was turned off of biology as an undergraduate. So the pictures that you see of a bacteria are cartoons. There are no organelles, they're very small, and you get this impression of it just being a Ziploc bag full of stuff. Well, really that can't be true, and there must be a lot going on in the bacteria cells that scientists purely could not see prior to the past 10 or so years. The second thing is that it's just a great challenge. So there's people looking at prokaryotic cells and eukaryotic cells. The eukaryotic cells are typically about 10 times bigger than the microbes. And really the diffraction limit of light, the limit at which the best confocal microscope that you could buy downstairs could do is on the order of the size of the cell. So bacteria cells really beg for this super resolution capability. So at Stanford, I worked on MREB superstructure. MREB forms a helix that gives the cell its shape. In my lab now, we're working on outer membrane proteins in a gut symbiont that digests complex carbohydrates, as well as localization of agents of virulence in Vibrio cholerae. But today, for the sake of telling a whole story, I'm going to leave these all behind. One very exciting structure that we finished up looking at just recently is FtsZ. And FtsZ, the important thing about it is it's the first protein, or one of the first proteins, that goes to the midplane of a bacteria cell, helps it constrict and divide. So this is very important for the perpetuity of the cells. And the Z-ring that FtsZ assembles into is still under debate. It's actually a hot topic. And I've been trying lately to figure out if it's a hot topic because it's such a cool structure to image, because it forms these nice rings, or because it's an important biological question. And I think there's a bit of both. And I think that methods in biology have to feed back and forth on each other. What we did to start looking at FtsZ was use dynamics as the active control mechanism. So we have this FtsZ protein. It's been bound by genetic fusion to EYFP, to a fluorescent protein. And when we look at the cells, every time we see fluorescent spots, we know that's FtsZ bound to EYFP. Now, there's something special that we haven't talked about yet today, and that has to do with how we're imaging this. We're imaging on, as Callie called it, a very fast EM-CCD. And these are really fast compared to the CCDs used in other places, but the integration times are between, generally in these experiments, 15 and 100 milliseconds. Now, on that time scale, because we have a pixelated detector, so this shows 3 by 5 microns, if we have a molecule that moves across, let's say, a few microns very quickly, we won't see it because it will be averaged out many, many pixels. So actually, in order to achieve high localization accuracy, to see these nice punctate spots, we need molecules that are stationary on the time scale of the imaging. So in this case, we're using 15 millisecond camera integration times, about as fast as we could push these instruments. And we see a few punctate spots. So as a guide to your eye, this blue is the cell, and these are spots of FtsZ at the midplane of the cells. And any molecules that are free in the cytoplasm are not resolved. Those can be picked up if we run a little bit faster, as was shown by Soyoung Kim. So at this time scale, we only see polymerized molecules. And it turns out that FtsZ works perfectly for us because it seems to be polymerizing and depolymerizing on the scale of 100 milliseconds. So now rather than using photophysics, we can actually watch a molecule polymerize, turn on, depolymerize, turn off. A different molecule polymerizes, turns on. So we can localize many molecules in that same very small volume at the midplane of these Caulobacter cells and see this turn on, turn off based on the dynamics. So I started my work in single molecules thinking, what a pain in the butt to try to figure out how to use live cells. And now it turns out live cells are actually working for me, and they're much nicer than fixed cells for this purpose. So we can watch the cells over time, and these are very fast frames, and we see some molecules, and they're blinking on and off, so that blink is characteristic of a single molecule, and we can localize them over time. And we can build up maps, so each of these cells, the orange, is the localization events. And we could see that the FtsZ is localizing to the center of the cells on a high resolution. And here we had about 50 nanometer resolution. Well, we could go one step further. We could say that was working, but not so well. We know how to do this active control trick. So now, instead of using YFP, we use the FtsZ Dendra 2 fusion. This is a photoactivatable fluorophore. And so now what we could do is actually turn on a sparse subset of FtsZ Dendra 2, image the red fluorescent proteins as they polymerize and depolymerize, so we get a few of these blinking events, and then repeat, turn on a new subset. And we get higher localization for two reasons. One is that we just get more events because of this extra switch, and the second is that Dendra is a redder fluorophore, and we get a lot less background from the cells. So this is a typical wide field image of Caulobacter cells. You could see all these cells and their crescent shape, and you see density of FtsZ at the center, as has been observed in biology labs previously. With super resolution imaging, we can now find them very, very precisely, and so we can see most of the cells have this nice stripe. It's very narrow, and before dividing, here's a cell that's about to divide, there's a focus of density. And we can look at these and see that this stripe is about 70 nanometers big, much wider than our resolution here, which is about 30 nanometers. And as the mass constricts, we see a slight thickening, but basically the density is getting very compact. We can carry this one step further and do some 3D imaging. This is a less sophisticated technique than the double helix that was mentioned previously. This is using astigmatism. Into the microscope, we introduce just one single optic. So a normal experiment, we see the emission through the objective, and it is collected by the imaging lens and projected onto the MCCD. A single cylindrical lens introduces an astigmatism such that on focus, there's a punctate spot as before. When you come out of focus in one direction, there's a horizontal astigmatism, and when you come out of focus the other way, there's a vertical elongation. So now we can fit this point spread function again. The center position of any of these functions is still the XY position, and the aspect ratio gives us the Z depth. So here we see an image of many Caulobacter cells. This is the same area. It's fluorescent. So every molecule you see is at the center of one of these cells. And what you see now is, unlike the nice single molecule images that you've been seeing, these are not punctate spots. They're actually elongated horizontally or vertically. And over time, we can actually see many molecules. This is using that combination of polymerization, depolymerization, and photoactivation, not seen here. And we could see all these molecules. So over time, we could localize many, many molecules. And we could build up maps. So whereas before, if we looked at a stock cell, we had many localizations. So here, to guide your eye, the cell is along this axis. Now we can actually look in Z direction and see this beautiful ring. The scale bar here is 200 nanometers, and our accuracy on the Z direction is about 100 nanometers. In a pre-divisional cell, just before dividing, we again see this tight focus, and when we look in Z, we also see a focused mass, perhaps a little bit spread out. And we can actually just cut this any way we want, because the data we have is simply a spreadsheet. And we can look at the molecules, the cells as they turn. And so the long axis of the cell is going from this way to into the plane of the board, and we see the structure of this ring. It's symmetric, it's the size of the cell, and most importantly, it's thicker than just a monolayer of proteins around the edge. So it's consistent with a model of proteins that are polymerized and that extend into the cytoplasm in some cooperative way. So let me now switch gears, and use the rest of my time to tell you about some really new work that's going on in the Myrner lab. And what we've been doing, in the Biteen lab. Unfortunately, I'm not privy to the newest work in the Myrner lab anymore. What we've been doing is looking at metal organic framework materials, so switching completely to the solid state. And this project is a collaboration with my colleague Adam Matzker, who's also in the chemistry department. And so, for example, if we look at these materials, UMCM1, which is University of Michigan crystalline material 1, these are made up of metal centers. So here's a zinc cluster at the center and different types of organic linkers. Here we have the tritopic BTB linker and the ditopic BDC linker. So units like that come together to form microporous cages and finally mesoporous channels in the whole crystal structure. And the exciting thing with these materials is twofold. From a practical standpoint, these are microporous materials with exceptionally high porosity and surface area. So the surface area here is over 5,000 centimeters squared per gram. So this is a large, large area that doesn't even mean anything to me yet. But the point is that it is very useful for catalysis, for separations, for gas storage and hydrogen fuel cells. So we can think about lots of things to do with these metal organic framework materials. Now, what I think is super exciting about it is that they're really small. And so we have these features on the nanometer scale, these pores that are about 3 nanometers, and these cages that are about 2 nanometers. And all that's known about them is from a crystal structure. So we have a predicted structure, we have an average structure, but we don't know the nanoscopic data. And so that's what really excites me about it. So we can make different types of metal organic framework materials, or as they're called at University of Michigan, microporous coordination polymers. This is a subclass of MOFs. We could change the linkers or change the ratios. So this material here is called UMCM2. It only has BTB linker, it has BTB and TDC linkers now. And this forms only these cages, but no channels. And a whole class of materials have been invented at Michigan. So this UMCM1, as I discussed before, has linear channels. UMCM2 has these just cages. And then UMCM4 makes planar layers without any barriers within those planes. And I'll talk to you a bit about what diffusion characteristics come out of that. So in general, there's many applications. And what we're interested in seeing is what does a guest molecule do when in these materials? And in particular, do the bulk characteristics really dictate the behavior? So the way that these materials are characterized in the bulk is by X-ray diffraction to get the crystal structure, by nitrogen absorption to get the percentage porosity, and by argon absorption plus some modeling to get pore size distribution. But clearly, it seems intuitive that the crystal structure is not everything. And if we're thinking, for example, of catalysis, Callie was showing us gorgeous examples in the side of SIRS, where every little spot is subtly different. And if we think about catalysis, that has to be true as well. So what we did was we imaged Nile red inside of these materials. As W mentioned before, Nile red is very surface sensitive and very environmentally sensitive. So the nice thing with Nile red is we only saw molecules that were inside the crystals and not the others. This work was done by Yi Lau in my group. So Yi incubated the materials in Nile red and then pre-bleached them so that there wasn't too much background, partly from these leftover metal centers that could be there from the synthesis, and partly just from having too much Nile red. He then waited 2 to 5 minutes for new Nile red molecules to diffuse into the crystal. And what he pointed out to me that was very exciting here is that experiments that have previously done on porous materials with fluorescence had the fluorophores baked into the synthesis. But here what we're doing is actually introducing guest molecules that will really mimic things like storage, where the molecules have to come in from the outside. And then the imaging is done at 561 nanometers and 29 hertz. And so when we look at these crystals, they form these nice long needles due to their linear channels. And we can image diffusion in the crystals. And here we start to see the molecules moving back and forth along. And so we see predominantly 1D motion with a few other subtleties. We can look a little more in depth at the tracks. And so here I'm showing only trajectories that last half a second or longer. And there's a few different modalities that are seen. So this red one here is a primarily linear track. And so if we watch it here, we see this molecule that's going up and down. It's moving in 1D, but you also see that it's jumping somewhat from channel to channel. So we see this structure where there's motion along channels, but also maybe some jumping to other channels. And this black is showing us a region where, for a small amount of time, there's some confinement. On the other hand, if we look at this pink arrow, this trajectory here, we see a molecule that's confined, jumps, is confined, jumps again. And so this is a molecule that's doing some sort of hopping between channels. And we think what's happening is these channels have dead ends. So you can see, for example, here there's a nice long channel, but there's an end at either end. And so the molecule goes back and forth until it somehow manages to find its way out. And it can find its way out through these mesoporous cages. Finally, down here, this blue molecule, which is just a tiny dot, is showing us a confined particle that is not moving over the course of the image acquisition. In UMCM2, on the other hand, there's no predicted channels or planes. So the only molecules that we see end up being trapped. They diffused very slowly into the material, and they are trapped for some time. So we see this movie where every molecule we see is trapped except a few, and there was one running around there. So we can track them now, and so if we look at this blue molecule, it's tiny, it barely shows up. This is what we generally see, these very highly confined tracks. However, this red track over there, that was one that probably jumped out in your eye previously, is moving a lot. And so that seems to be diffusion in a defect void. And so single molecules can actually show us something about the proportion of defect voids. Finally, in UMCM3, we see planar diffusion along these planes here. And so we see long trajectories. The molecules can't come out of focus once we find them in focus. And they're diffusing freely in 2 dimensions. Most of the molecules have this free diffusion, whereas, again, we see some highly confined molecules. So what can we do with this quantitatively? Well, we can think about them in terms of the mean squared displacement. And single molecules lets us take every single trajectory, calculate a mean squared displacement. And as we know, the mean squared displacement, or the average R squared, is linearly related to the diffusion coefficient. And n gives us something about the dimensionality of the motion. So we could do that for UMCM1, and we see some tracks. UMCM2, we see some, and UMCM4. But 3 things jump out. Number 1, they are not homogeneous. And so in both cases, you see some red curves that have a positive slope. Those are mobile molecules, as well as blue curves that are pretty much flat sloped. Those are immobile molecules. Number 2, there's a different distribution. So UMCM1, for example, has many of these red tracks and few of the immobile tracks, whereas UMCM2 has many more immobilized tracks. And finally, if we look at the slope, it doesn't end up giving us n. It doesn't end up agreeing with 1 or 2 for 1D or 2D motion. And heterogeneity, again, is masked because each of these, as powerful as it is that each one is one molecule, if we look at a track like this, there's a lot of free motion, but then there's some confined motion that I've highlighted with that black square. So to treat the heterogeneity, we want to use a diffusion model that accounts for multiple diffusion coefficients and multiple types of motions, actually using the single molecule data. And so we want to use the cumulative probability distribution, or the probability that the squared displacement will be bigger than some given value in some time period, to really look at what's going on. Now, the cumulative probability distribution depends on the dimensionality. There's an equation for 1-dimensional, and there's an equation for 2-dimensional. So we first need to know the dimensionality of motion. We look at the directionality by doing an angle analysis. So for every step, if we go from 1 to 2 to 3, we measure angle theta. If theta is 0, that means we're moving straight forward. If theta is 180, we're moving straight backwards. In UMCM1, with its linear channels, we see the following distribution. Most of the motion is either 180 degree or 0 degrees, so the motion is linear. It's mostly moving forward or backwards. We do one more thing. We reduce the quasi-1D motion to true 1D motion, so we project all of these trajectories against their backbones to really have pure 1D motion in that case. For UMCM2, which mostly had confined motion, we actually see a distribution of angles, but this is actually the distribution that's characteristic of a Gaussian error in localization. So because if a molecule is right here, it could be localized there, 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 there. As we move further from the center, there's more chance that the next step will be closer to the center. So these are very small steps, but they end up giving rise to this distribution. So we're not going to think about UMCM2 anymore in this motion analysis. Finally, UMCM4 shows average, pretty much no preference for any given angle. And this is traditional for 2D motion, where the diffusion could be in any direction. So we analyzed UMCM1 according to the 1D cumulative probability distribution. A nice thing we could do with the CPD is put in several terms. So here we have one term, two terms. They look about alike, but alpha and beta are weighting coefficients, and these can have different diffusion coefficients that come out of R squared, out of the MSD, by that Einstein equation. And then we also have an immobile term, such that all these components add up to 100%. So we fit for different taus. Here's a representative short tau, medium tau, and long tau. We could fit these CPD functions and get at alpha, beta, and R squared 1 and R squared 2, the MSDs. And we plot MSD as a function of tau. So here for the fast motion, we get a diffusion coefficient. For slow motion, we get a diffusion coefficient that's about an order of magnitude slower. So these are both linear. Neither are immobile. One is moving quickly and one is moving slowly. And then 5% of the molecules are left over as immobile, which agrees with the qualitative data. And then we can do the same thing for the 2D case. Here, our function looks a bit different. But again, we have three components. Fitting an immobile component in this case did not work. So we fit three different components. And what we assumed here was that we'd have three MSDs, a fast MSD or a fast diffusion coefficient, a medium one, and a slow one, and that the slow diffusion would be essentially immobile molecules, but we weren't sure exactly what to expect. So we fit many of these functions again, and now because this model is different, there's some interesting features like the shoulder. And now we can plot MSD as a function of tau for the three different cases. Now remember, it doesn't matter here if it's one molecule that starts off slow, then goes fast, or two totally different molecules. The blue is the fast trajectory, and it has a diffusion coefficient that can be quantified. The yellow is the slow component. Again, this is nice and straight, so it gives us, again, a diffusion coefficient that's, again, about an order of magnitude smaller. The third component was really interesting, though. Here it looks basically like 0, which makes sense for a very slowly moving particle. But if we blow it up, we see that it's not linear. It actually has a curved shape. It saturates. And this is consistent with confinement to a square with length L. So we can write out this equation where D naught is the diffusion coefficient and L is the square size. And by fitting our data to this equation, we get that there's very slow diffusion, another order of magnitude smaller, and that the lengths of these features are about 70 nanometers. So this is confinement to an area that's quite small, but actually bigger than our resolution, which here is 20 nanometers. So in conclusion, what we've done is looked at the diffusion of individual Nile red guest molecules inside this crystalline microporous coordination polymer host. And we looked at it in real time, and we're able to really get at the heterogeneity. There were several subpopulations of molecules. And trajectory analysis really allowed us to quantify these different modes of motion, as well as the percentages of molecules in each mode. Going back to the FtsZ story, what we were able to do was get the FtsZ structure in live cells in 3 dimensions using single molar fluorescence plus an astigmatic lens. Our resolution was about 30 nanometers in the lateral direction and about 100 nanometers in axial resolution, though I should point out that the diffraction limit in Z is much bigger. It's on the order of 2 microns, and so 100 nanometers actually was a surprise. What I was most excited about here, and what we're continuing to use in my lab a lot, is dynamics in live cells as our mechanism for active control. And finally, we were able to actually elucidate some biology, and so we saw the Z-ring structure did extend. It was overlapping filaments that extended away from the membrane, and that there was some thickening upon Z-ring constriction. So let me finish up by acknowledging everyone. The work on FtsZ was done in the Murder Lab. I'd like to thank WE. As I mentioned before, he was a terrific mentor and really made it a terrific environment for doing good science, while at the same time teaching us to pay attention to details, to really be careful. This scale bar was wrong until WE pointed it out the night before we submitted it. A lot of attention to details, and this was a very valuable lesson for me, as well as doing very exciting science. The work on FtsZ was done in collaboration with Aaron Goley, who is now a faculty member at Johns Hopkins, and Lucy Shapiro at Stanford University, and funded by the NIH. The work that I showed you in UMCM1 was primarily done by Yi Lau, a grad student in my group. It was actually begun by an undergrad in my lab, John Yang, and other people in the Biteen lab to help. This is another very, very nice collaborative environment where the people doing tracking of bacteria cells help the people doing imaging in solid state materials. The materials were made under a grant from DOE, and my work was funded by a Burroughs Wellcome Award. And congratulations, WE. Thanks.
Again, we do have a few minutes before the break here. If anyone has any questions for Julie.
Yeah. That's a great question. There seem to be some, and we actually played a lot. Actually, there was some, because we played a lot with different solvents. Partly because the diffusion coefficient is obviously affected by the solvent, and partly because the fluorescence intensity of the vinyl red is very much affected. So those were done in butanol, and actually something I didn't mention is that these UMCM1 movies were done with 10% mineral oil, because the motion is so facile that we had to slow it down a bit. Yeah, absolutely. So we actually, every single experiment we do, we play a lot with it. The videos where I pointed out the frame rate, the 15 milliseconds, those were doing YFP and FtsZ, and that was actually me going as fast as I possibly could while still being able to localize a fluorescent protein nicely. Obviously, there's a trade-off always between time and space, so number of photons will decrease. The 3D experiments were actually done at 50 milliseconds, so they were slowed down a bit because I just wasn't getting enough signal to background. In these materials, Yi played a lot with the timing. 100 milliseconds, he found he wasn't getting long enough trajectories. There wasn't enough data. Going down to 15 milliseconds, he just wasn't as happy with the signal to noise. And I think a lot of these things, like being able to see very subtle things like hops from channel to channel, you really want to be getting this 10 nanometer scale accuracy rather than accepting a mere 40. Yeah, it was. And so it was partly the surfaces of the channels, and we still haven't thought too much about the real chemistry there, but partly also just the solvent made a big difference. So the hydrophobicity in the channels can also be affected by the solvent. So it worked. And the nice thing was we did not see, if we go next to the crystal, those molecules that are just free in the solution are very dim. So that was really nice not to be, as the PI, me sitting there saying, are you sure those molecules are in there? Especially when we see the anomalous motion, we really want to make sure they're inside the crystal. So yes, it seems to work, though it wasn't 100% predicted before. Yes, that's actually how they got my attention. So in Adam's group, Dr. Koh, who's the main guy for the UMCM materials, he had taken one of these UMCM crystals, so they're these nice long needles, and he put the dye it's actually not localized, it's just he put it on top of the material, and you could see it starting to come in from the edges, because most of the motion, the fast motion, is definitely along the channels. So they know that's there. And you also, interestingly, even in this bulk experiment, could see some slow diffusion this way. And you could imagine characterizing the rates and things like that. Though what I think is more interesting is thinking about the rates as a nanoscopic thing, because I think a lot of this motion coming into the crystals is probably through defects rather than through the pores.
Okay, we're going to start our break now. I think if you have any more questions, please say any of the speakers, and please be back here at 4:10 for our last two talks. Thank you again.